All right. Well, hello, everyone. Welcome to another session of our Evolution Seminar Series. Today, we'll be digging into phylogenetic networks as we are joined by Kevin Kong from the Solis Lemus Lab, working in the Wisconsin Institute for Discovery, doing really fascinating work, helping us understand evolutionary relationships. So without further ado, let's get right into it. Join me in welcoming Kevin Kong.
All right. Hello, everybody. Thank you, Jashim and Emily, for organizing this evolution seminar. And as Jashim just mentioned, my name is Kevin Kong, but my real name is Sungsik Kong. So please feel free to call me Kevin. The reason that I named myself Kevin was when I moved from South Korea to North America for my education. It was Christmas time, and I watched a movie called Home Alone the night before, and someone was asking me my name, and I said, "Oh, my name is Sungsik," and they said, having little trouble pronouncing my name, so I said, "Kevin is good," because that was the only name I could remember. So Kevin is good. I'm a postdoc at the Wisconsin Institute for Discovery. I work with Dr. Claudia Solis Lemus, and I work on phylogenetic networks, which is an extension of what the phylogenetic tree, extension model of the phylogenetic tree. So I heard most undergrad students here are majoring in genetics. Is that right? Okay, so a lot of content in my talk will be very methodological, but I also did my undergraduate and master's in biology. I majored in zoology and evolutionary biology at the University of Toronto. And I jumped into more method stuff for my PhD at the Ohio State. And today I'm going to spend some time talking about some general overview of phylogenetic networks, which is kind of a new concept. And also I'm going to talk about what's going on behind our novel method called PhyNEST. It's an acronym for phylogenetic network estimation using SiTe patterns. But if you are confused in the middle of my talk, please feel free to stop me and ask what am I talking about. Okay, so I will start. Okay, good. So I do phylogenetic method development, which means I work with this structure all the time. This is a phylogenetic tree, and I show a very simple cartoon example of a phylogenetic tree that contains 5 species. And each of the species are labeled here on the bottom. And this is a rooted phylogenetic tree, which means it has directions. And the beginning point of this rooted phylogenetic tree is the root on top. That represents a single point. It represents the most recent common ancestor of all of the species under consideration. And in between the root and the tips or the leaves, we have a series of internal tree nodes where a single branch splits into 2. It represents an ancestral species that gave rise to all of the descendants below that node, but it also represents a speciation event, meaning a single ancestral species gave rise to 2 descendant species. Is this kind of intimidating you? So, okay, I'm not going to use it. Yeah, so internal tree node represents speciation event as well as the ancestral species. But the question is this phylogenetic tree a perfect model to depict all of the biological processes that have shaped today's biodiversity? The answer is unfortunately no, because phylogenetic trees often overlook one of the very important biological processes that also can result in the production of a novel species, like hybridization, that refers to interbreeding between 2 distinct species, and sometimes it can also result in the production of a novel species. And a lot of people ask me, is hybridization a thing? Is this actual biological process? And historically, it has been seen as something unnatural, and even if it somehow happens in nature, its offspring is not expected to persist over evolutionary time. So we can simply ignore the presence of hybridization in previous biological investigations. But thanks to the accumulation of genomic data and the computational methods to understand this hybridization, we now know that hybridization is much more common in nature than we previously thought. And some studies tells us that 25% of plants and 10% of animal species probably have experienced hybridization at some degree during their evolutionary history. One way to conduct a phylogenetic analysis, even though we know hybridization is going on, is simply we ignore the presence of hybridization and stick with the tree analysis. But such practice, unfortunately, will lead to a biased species tree estimation, which means no matter how much time and computational resources and how much data you have, the resulting phylogenetic tree is going to be wrong. It's not going to tell us the true relationship between the species, and the interpretations made based on that biased tree will be misleading, and we don't want that. So to overcome this problem, a concept of phylogenetic networks has been proposed. And phylogenetic networks and trees are very similar, as you can see. They look very similar. And they are both composed of a set of nodes and the branches that connect those nodes together. But the networks are distinguished from the trees by this reticulation structure, which extends the tree model by allowing two branches that are once separated to be merged back together, creating a reticulation structure. And it has a reticulation node, which is marked in pink circle over there, that has two incoming branches and one outgoing branch, denoting a hybridization event. So here we can say species 4 is the hybrid daughter of species 3 and species 5. But in practice, there are two types of networks are being used in biological investigations. And these two networks are abstract network, which an example is shown here on the left side, and the explicit network shown on the right side. And two networks look very different from each other. And as much as they look different, their algorithm for reconstruction, as well as the biological interpretations you can make based on these two networks are very different. And to be more specific, abstract networks typically result in colorful diagrams because their tips are usually shaped in circular shape, and they are colored using some sort of external information, such as the geographic location where the sequences were sampled. And these abstract networks are usually large and very complex in structure because they are fast to compute, which means the data set can have large, many number of sequences, and it has many cycles in its structure. So it looks very complex. It seems like something must be there. But relying on the reason that they are fast to compute is because they use distance matrix to build one, which means they assume phonetic philosophy, which is it assumes two sequences are evolutionarily related with each other when they are similar in terms of their nucleotide arrangement, which is not always true in nature, because even though two lineages take completely different evolutionary pathway, it can result in similar sequences through processes like homoplasy that includes parallel or reversal evolution. Many cases, abstract networks are unrooted because it lacks the rooting process, which means we can't identify the ancestor-descendant relationships between the nodes, which is probably the most basic information that you want to get out of the phylogenetic analysis. So combining these 3 points, the point for abstract network is clear. It's not designed to understand evolutionary relationships between sequences or species, but it's designed to display signals in data. In particular, it visualizes conflicting signals using cycles, unlike trees. On the other hand, explicit networks looks very similar to the trees, very similar to the phylogenetic tree that we know of, but it has a number of horizontal branches that denotes an hybridization event. And because estimating these explicit networks are model-based, they're computationally heavy, which means usually the number of tips it contains in the explicit network is small, which means we conduct explicit network analysis using relatively smaller data set than the abstract networks. Many cases, the explicit networks are rooted, meaning we can understand the directionality of the evolution. But in some cases, people also use what's called semi-directed explicit network, which means it doesn't have a directionality among the tree branches, but it has a directionality among the reticulation branches. So at least we can know who are the parents and who are the offspring in the hybridization event or in reticulation structure. So the point of explicit networks is to infer complex evolutionary histories like hybridization on top of speciation events that is usually inferred in phylogenetic trees. So now I hope I made it clear that abstract networks are useful to visualize signals, and explicit networks are useful to understand how different species experienced complex evolutionary processes in their past. But unfortunately, in practice, people use abstract network on empirical data sets, and they try to make some biological interpretations based on abstract networks. So here I highlight one of the papers that was published in the beginning of the pandemic, April 2020. When the lockdown just began, and then I was stuck at home, I was working on my dissertation, and a paper with a very ambitious title was published in PNAS, which is one of very prestigious biological journal we have. The title of that paper was Phylogenetic Network Analysis of COVID-19 Genomes. And it was very exciting at that moment because we virtually didn't know anything about COVID, and somebody's doing phylogenetic network analysis of this COVID. I thought this will help us to combat the pandemic and end this lockdown faster. In this paper, they had a single figure, which is a median joining network shown here, which is a type of very popularly used abstract network. And based on this figure, the authors tried to make 2 points. First, they tried to identify the origin of the pandemic. Where is the beginning point of all this COVID-19 pandemic? And also based on this figure, the authors tried to trace the entire transmission history of the virus that has been spread internationally at that point. And myself, along with my colleagues, including Santiago shown here, we had to write a rebuttal letter to PNAS with a very strong title to make things as clear as we can. We say, okay, median joining network analysis of COVID-19 genomes is neither phylogenetic nor evolutionary. And the point was, because median joining network is a distance-based method and it doesn't have a root, we can't understand the directionality of the evolution, which means we can't understand who is the ancestor of all these viruses. So we can't identify the starting point of the pandemic. But also, even if it somehow magically identifies a true evolutionary relationship between COVID-19 genomes, relying on a single phylogenetic tree or a network to understand the entire transmission history is not a good practice. So what should have they done? They should have inferred an explicit network. And in the past 20 years or so, computer scientists, mathematicians, and statisticians, as well as biologists, have been working hard to understand the structural properties of these explicit networks. And today we have more than 20 subclasses of explicit networks. And each of these, the relationship among these subclasses are shown here in diagrams, where each of the nodes represents different subclasses, and they are linked based on their structure similarity. This is great, but a lot of cases, the reason the new subclass of explicit network was proposed is to understand their mathematical and structural properties like k-nested networks here, which is a little bit tricky to infer and also to make biological interpretations based on these networks. But I'd like to highlight 2 subclasses of this explicit network that has been very useful for biological investigations. And the first one is trees. And yes, this is the phylogenetic tree that we've been all working with. And I say trees because it's a special case of explicit networks where the number of reticulation equals to 0. So the structural characteristics of the trees is no reticulation. But more interesting for us network people, we are more interested in understanding the level 1 network, which is very closely related to the trees. And the structural property of level 1 network is the reticulations in the topology are isolated with each other, assuming the hybridization event occurs independently all the time. So a lot of computational methods that estimates phylogenetic networks from genomic data assumes the true relationship among the species can be described using these level 1 networks. And as I said, in the past 20 years or so, a handful of computational softwares or handful of methods has been proposed to estimate phylogenetic networks from genomic data. And they can largely classify into one summary-based methods and full data methods. The summary-based methods take two-step process. We have raw sequence alignment, and we first summarize this sequence alignment into a set of gene trees, which is the evolutionary history for a single gene. And we use those estimated gene trees as input file for network estimation. The advantage of this summary-based method is that they're relatively fast because a lot of hard work has been done in prior to the network estimation. But the downside is that the quality or the accuracy of the final phylogenetic network completely relies on the quality of estimated gene trees, not the quality of the sequence data or the algorithm itself. And to overcome this problem, we have different approach, which is called full data method, which directly takes sequence data as input and bypasses gene tree estimation step. The advantage is it uses all of the signal in the raw data and it utilizes all of the phylogenetic variability in the data, but the downside is it is computationally very heavy, limiting their use to a very small data set. And there are some examples of computer softwares that can do this for summary-based and full data methods. But in this talk, we're going to talk about a method called PhyNEST, which I proposed last year, which tries to overcome the limitation in summary-based and full data methods. Okay, so I will end the general overview of phylogenetic networks here. Anybody has any questions before we jump into how PhyNEST works behind the software? Anybody from the online? Are we good? Okay, good. All right, so this was the state of computational method for estimating phylogenetic network when I began my PhD dissertation. Of course, PhyNEST wasn't there, but this was a state when I began about 5 years ago. And the main question I had for my dissertation was very natural and clear. Can we estimate phylogenetic networks directly from sequence data, bypassing the gene tree estimation step efficiently, as efficient as the summary-based method and accurately as accurate as the full data methods? And what do you think the answer would be? Would it be yes or no? Anybody? The answer is yes, and that's thankfully how I finished my dissertation. And we do so by combining what's called SiTe pattern frequency data that you can extract directly from the sequence alignment and composite likelihood framework. So what do I mean by SiTe patterns? SiTe pattern is an assignment of nucleotide states at the tips of a topology. So let's consider an example topology shown here. Again, contains 5 species and a single hybridization event and the corresponding sequence alignment on the right side. If we select the very first column of the sequence alignment and we map that onto a topology, we get the SiTe pattern of AAAAA that tells us there's no variation between the 5 species at that specific site. We can do the same thing for fifth column of that alignment that represents site 5 of the 5 species under consideration. And we map that onto a topology, we get the site pattern of TTAAT, which tells us a bit more story than the previous case. Here, species 3 and 4 shares the same nucleotide that is different from everybody else. And at this point, you might wonder, hey, this sounds very similar to the SNP data. What's the difference between site patterns and the SNP data? So for insight patterns, we assume every site, we call every site is coalescent independent site, which assumes every site has its own evolutionary history. And we also make use of those sites where no variation was observed, like in site one here. Whereas the SNP data, the biggest difference would be they will only consider those sites where the variability was observed. So site patterns in the SNP data, they're two different things, even though they might sound similar. So for the next few minutes, I'm going to talk about how we compute the composite likelihood of a network here using site patterns. So again, here I show a cartoon example, 5 species single reticulation. But before I do that, I want to formally introduce a number of parameters that we are interested in estimating. The first parameter is tau, which is a vector of speciation times or node ages, where each element in that vector is assigned to each internal tree nodes. And these node ages are measured in coalescent unit backward in time, meaning its value is 0 at the present time, and the value increases as you move towards the root. And it represents the timing of the split event or timing of the speciation event. The second parameter that we are interested in estimating is gamma, which is also called inheritance probability, hybridization parameter, parental contribution probability, and so on. And it is assigned to one of two branches towards the reticulation node. So here we have a reticulation node. We have two branches going towards the reticulation node, and the gamma is assigned to one of them. And because gamma is a probability, its value ranges between 0 and 1, which in turn makes the inheritance probability for the other incoming edge as 1 minus gamma. And of course, gamma has some biological meaning, which can be interpreted as the proportion of genomic contribution from one of the parents toward the hybrid daughter. And finally, we have another parameter called theta. It's a population size parameter. And for computational tractability, we assume that there is a single constant population size throughout the entire topology. So in order to compute the composite likelihood of a network, the first thing that we want to do is we want to break the network down into a set of displayed trees. And we get displayed trees from a network by removing one of two reticulation branches at a time. And here, and each of the display tree will also have the probability, which can be computed by multiplying all of the gammas that are assigned to the reticulation branches that are not deleted in the production of particular display tree. So here, our network N is decomposed into two display trees, where display tree 1 has a probability of gamma, and display tree 2 has a probability of 1 minus gamma. And for each of these display tree, we further decompose it down to a set of quartets, which is simply a phylogenetic tree that has 4 tips. And we do so by selecting 4 leaves from the display tree, along with some of internal tree nodes that represents most recent common ancestor for the pairs of taxa, as well as the branches that connects those nodes together. So here, by selecting species 1, 2, 3, and 4, along with some of the nodes and the branches, we extract our first quartet from our display tree 1 and repeat this process for every combination of 4 to extract all of the quartets extracted from the display tree. And of course, we repeat the same thing for the rest of the display tree. So from our network N, we got our 10 quartets extracted over the display trees. And each of these quartets will also have the probability that is assigned to the display tree that the quartet was decomposed from. Now we're going to zoom into one of these quartets. And since at a given site, each tip can have any one of 4 nucleotide states, A, C, T, or G, which means in this quartet, there can be 4 to the 4 or 256 possible site patterns. And a good news is these 256 site patterns can be displayed in this flattened matrix, which is 16 by 16. And even better news is we can compute the true probability of any one of these site patterns under a quartet under the Jukes-Cannon model, which is a simple nucleotide substitution model. And even better news, make my life easier, is that under the Jukes-Cannon model, some of the probabilities of these quartet site patterns will be equal to each other, which means we can generalize these 256 site patterns down to 15 patterns shown here. So letting I, J, K, and L denote distinct nucleotide states, A, C, T, or G, these 256 site patterns can be generalized into 15 site patterns shown in the second line here. And we're going to call this vector as PQ. And as I mentioned previously, the true site pattern of any one of these 15 generalized site patterns can be computed under a quartet tree, given the set of speciation times, as well as the population size parameter theta, as shown in Schiffman and Kobatko 2015. So, given that we can compute the true site pattern probabilities of any quartet tree, we can also compute the true site pattern probability of any quartet that is extracted from a network. So let's say we extracted a quartet from our network and that has species 1, 3, 4, and 5. We decompose this network quartet into display trees. We compute the true site pattern probabilities for for these two quartets, and we compute the weighted sum of these true site pattern probabilities for these two quartets from each of the display tree to get the true site pattern probabilities for the network. And by weight, I mean the probability that is assigned to the quartet or the display tree that the quartet was decomposed from. Now we can compute the true site pattern probability of any quartet tree or a network. And in order to compute the likelihood of that quartet, we need observed site pattern frequencies, and we get this from our data. So the idea is we select 4 sequences from the alignment where the 4 sequences also occur in our quartet of interest. And focusing on these 4 sequences, we start from the very first site. We see the site pattern in every site. For example, the site in site 1, the site pattern is AAAA. We decide which one of 15 generalized site patterns it belongs to. And we cumulatively count how many times each one of these site pattern occurs from the beginning to the end of the alignment. So at the end of this process, we will have a vector of length 15, where each element is an integer, and some of those integers will be equal to the alignment length. And we're going to call this vector WQ. So having these true site pattern probabilities that can be computed for any quartet tree or a network PQ, as well as observed site pattern frequencies WQ, we can compute the likelihood of a quartet. And by multiplying these quartet likelihoods together that are extracted from the network, we can compute the composite likelihood of a network. At this point, you might ask, but Kevin, since the quartets are not independent from each other, are we really computing the full likelihood of a network topology? You are right. The quartets extracted from a network are not independent from each other. So by computing likelihoods through this approach, only approximate the full likelihood rather than computing the exact likelihood of a network. But fortunately, what's the good news is, even though we are approximating the full likelihood, getting the likelihood through this approach has been shown to be performing quite well in phylogenetic context, which is a good rationale for us to stick with the composite likelihood framework. And this is why, in many cases, composite likelihood is also called pseudo likelihood. Okay, so now we have a way to evaluate the fit of the data onto a topology. Our next goal is find the topology that describes the data best. And we do so by using heuristics. And to explain how heuristics works in network space, let's consider this landscape, a terrain that has 2 mountains. We're going to call the peak of the lower mountain local optimum. We're going to call the peak of the higher mountain as global optimum. And any point in these 2 mountains is a topology, and higher the altitude of that topology, higher the likelihood. So the goal of heuristics, we start at a certain point in this network space, we somehow traverse around, and we want to eventually get to the global optimum that represents a topology that has highest likelihood in the given network space. Of course, this is a very simplified representation of what the network space actually looks like, because every time we add a reticulation onto a topology, the dimensionality increases. And every time we move up the dimension, a whole new space of networks appears that needs to be searched. What's more complex is every time we move up the dimension by adding a reticulation onto a topology, the likelihood of the topology at the global optimum in that network space also increases. So unless we limit the number of reticulations in the topology in our final network, or unless we limit the number of dimensions of network space that needs to be searched, there will be 2 problems. Number 1, the search will go forever because it's going to find more complex topologies that's going to describe the data better. And number 2, even if it somehow stops based on some termination criteria, the final network will have excessively large number of reticulations, which will make our life difficult because it's going to be difficult to make biological interpretations when there are so many reticulations. Unfortunately, there is no straightforward way to identify the expected number of hybridization in prior to the analysis, although there are a number of ways to that after the analysis. And this is the place where the knowledge about the biological knowledge about the system in the data comes into very important play. So people like us who work in the methods and the biologists who are working with empirical data should collaborate to produce a network that makes biological sense. Anyway, to traverse the network space efficiently, PhyNEST have implemented two strategies. And first strategy is called hill climbing. The idea is very simple. We start from some sort of a starting topology which can be randomly generated or estimated, a tree topology estimated from the data. We compute composite likelihood of that starting topology, and we make a little tweak on the starting topology and propose a new topology. And we compute likelihood for that proposed topology. If the likelihood of the proposed topology turns out to be better than the starting topology, we're going to throw away our starting topology and we're going to move up in the network space and try to make a little tweak onto the proposed topology. And we repeat this process until we find the topology, hopefully in the global optimum, where no tweaks will improve the likelihood of that topology. The nice thing about this strategy is it's really straightforward to understand, and this is why they have been implemented in many of the tree or network inference methods. But the downside is that because it only traverses the network space by allowing the upward movement, it has risk of becoming stuck at the local optimum, which is another peak in our network space that is not the highest one. So to overcome this problem, we have implemented our second strategy called simulated annealing. So simulated annealing and hill climbing operates in a similar way. It tries to accept the topology that has better likelihood. But unlike the hill climbing, simulated annealing also makes downward movement by accepting the topology that has worse likelihood than the one before proposal at some probability. The advantage of this simulated annealing is that it decreases risk of becoming stuck at the local optimum and increases chance of eventually getting to the global optimum. But the downside is because it searches a network space much more exhaustively than the hill climbing, which can be directly translated into increased computational time. So simulated annealing will have increased computational time than the hill climbing. Okay, so any questions so far? Any question about the methodology behind PhyNEST at this point? Any terminology? Yes?
Hill climbing result is depend to the starting point, yes?
Not necessarily, but yes, yeah.
And how many starting point do you consider in your algorithm?
Right. So so the question was so depending on where we start the search in hill climbing, the risk of becoming stuck at the local optimum is high. So how many starting points do we have? Okay, typically the network analysis conducts 10 independent runs. It depends on the user, but 10 is the typical number. And at every run, we have two options. One is we fix the starting point of the search. And this is the case when the starting topology was estimated from the network. And we're kind of confident that this is a good representation of the topology. That's one option. Another option is we make a little tweak to the starting topology that is provided by the user to start the network search in multiple places. So we have two different options. Yeah.
Can you run into the problem where you have more than one local optimum?
More than one, yeah. I think it is very possible in the network space, and that's probably the little risk of relying on a hill climbing algorithm, but not always. Yeah, hill climbing also performs very good. I hope I don't sound like hill climbing is not useful, but it is also very useful. But we have another way. Anybody else? Okay. So all of these methods that I have described so far has been implemented in a computer software called PhyNEST. It's a Julia package. And I tested its performance of PhyNEST using simulation and empirical data. And here I show an example of the result from the simulation study. So the idea is we set the true topology looks like this. It contains 7 species. There are 2 independent hybridization events in the true topology. We simulated gene trees based on this species network over a wide range of number of loci, ranging from 100 to 5,000. And for each of these loci, we simulated sequence alignment. And using those sequence alignment, we estimated maximum likelihood gene trees using IQ tree. So the reason that we simulate gene tree and estimated gene tree again was we want to test those methods that requires a set of gene trees as input. So we use the simulated sequence alignment as input for PhyNEST using hill climbing algorithm and simulated annealing algorithm. And we use the estimated set of gene trees as input file for Phylogenetic and SNAC, which uses a set of estimated gene trees as input. We conducted 100 replications and we evaluated the accuracy of the networks that are estimated using this 4 method by comparing the estimated network with the true network. So here is the result from the simulation data for accuracy. So each of these points on the plot represents different method, different colors represents different methods shown here. X-axis represents the data set size. As you move left to right, the data set size increases in terms of number of loci. The y-axis represents the topological error measured using hardwired clustered metric. It's called something like, it's similar. It's basically identifying how different 2 topologies are. So higher the value, more different the estimated topology from the true topology. The first thing to notice, which is a good news, is for all of these 4 methods, the accuracy increases as we add more data, which is kind of expected, but it's nice to see that it is actually the case. And focusing on each of gene trees, number of gene trees, when the number of gene tree was 100, when there was small amount of data, all 4 methods did not perform very well because we needed more signal in the data. And in this case, PhyNEST using hill climbing algorithm turns out to be performing better than any of the 3 other methods. But when we add more data, when the number of loci increases to 500 and 1,000, all 4 methods performed quite similar to each other. But when we have large number of gene trees, 3,000 and 5,000, it became clear that PhyNEST using simulated annealing algorithm were more accurate than the other 3 methods. So this is a good news that PhyNEST is working quite well and reliably. What about the compute? Yeah.
Why you don't have a result for 500 size 100?
Hill climbing is right here on the bottom. So hill climbing, simulated annealing, SNAC, and Phylogenetic.
But for number of yes,
This one actually there are 2 points here. The accuracy of SNAC and PhyNEST using hill climbing were basically almost the same. That's why they were merged with each other. So it looks like there are 3 points.
difference hill climb and simulated annealing is significant based on your.
Yeah, it wasn't significant until number of gene trees were 1,000, but when we add more data, simulated annealing turns out to be performing better than the hill climbing. Okay, so at this point, it sounds like, okay, so PhyNEST using simulated annealing algorithm is the way to go. But does it have any disadvantage? As I mentioned, the simulated annealing takes much more time than the other methods. So this plot summarizes the computational time that is required to estimate networks. At this point, let's ignore these red bars here. So we want to focus on the other 4 colors, not the red ones. The first thing to notice is all of the methods are clustered close to x-axis, meaning the method, the Phylogenetic, SNAC, and PhyNEST using hill climbing are computationally very efficient. They were very fast to compute. But for PhyNEST using simulated annealing, which is represented in blue bar, it takes much longer time than the other three methods. And in average, it took about 20 hours to complete the analysis. So basically, PhyNEST using simulated annealing becomes more accurate in the cost of computational time, so computationally intensive. And now we can focus on this red bar here, which is time that is required to summarize the sequence alignment into a set of gene trees using IQ tree, which is one of the most efficient methods to estimate gene trees from the genomic data. And when the number of loci was small, the time that is required to summarize the alignment into gene trees were not really significant, but it increases as we add more number of loci. And when the number of loci was 5,000, it finally, the time required to summarize the sequence alignment into gene trees sometimes exceeded time that is required for PhyNEST using simulated annealing algorithm. So we are saying, okay, simulated annealing algorithm for PhyNEST takes a long time, but it seems like the summary-based methods are efficient, and in fact, it is efficient, but we need to consider the time that is required to summarize the alignment into gene trees to even initiate the summary-based methods. And when the number of gene trees is large, that time can be much larger than the simulated annealing algorithm. And of course, this process of estimating gene trees from the data can be parallelized, which will improve the computational speed, but that's only possible when you have such computational resources. And in many cases around the world, people rely on their local machine to conduct the network analysis. All right, so I also applied PhyNEST for empirical data set, and I selected a monkey data set shown here because I like monkeys, and these are my favorite animals. We have 7 species, and the gene flow among these 7 species of monkeys were shown in the paper Van der Poel et al. And the gene flow among these species were computed using what's called delta. It's similar to the D statistic, which detect hybrids using a set of gene trees. So one clear thing is, okay, the gene flow or hybridization event among these monkey species are rampant. And we estimated phylogenetic network using PhyNEST, using those 7 species plus an outgroup that is recommended from the original paper. We estimated the network assuming there is a single hybridization event, and we see that there has been a hybridization event among the monkey species that occurs in Africa. And when we increase the number of expected hybridization in the network to 2, we get our network that has 2 reticulations where the reticulation among the African species was retained, but we also additionally inferred a reticulation event among the Asian species. So in summary, I hope I convinced everybody that phylogenetic network is a thing. So everybody should know phylogenetic networks is very useful in evolutionary investigations. And I wish in the future, the field of phylogenetics and evolutionary biology shift from the reliance of the trees towards the use of networks to accommodate both speciation event and complex evolutionary histories. And if we want to use networks for our study, we must use explicit networks because they are the ones that tells us the evolutionary history of the species, not the abstract networks, even though abstract networks will give you more beautiful figures and it's going to finish your analysis much faster. And to do that, we propose a new method called PhyNEST. Again, it's an acronym for phylogenetic network estimation using SiTe patterns. And this method, this software is publicly and freely available in this GitHub repository here. And currently we take the sequence alignment in the format of phylib, and it yields phylogenetic networks using either simulated annealing or hill climbing searching strategies. And the biggest advantage of PhyNEST compared to the existing method is it bypasses data summarization step, which can take long time if you have large number of loci. And also there is a risk of diluting the signal in the raw data. And it utilizes all of the phylogenetic signals and variability in the sequence alignment. But is the PhyNEST perfect method? And unfortunately, there are a number of gaps. There are actually many gaps that needs to be filled to be applicable for a wide range of biological systems. And the first problem that I am tackling right now is to find the root position using SiTe patterns. So currently, because PhyNEST try to estimate rooted phylogenetic network, we require users to provide an outgroup sequence, which is an easy way to root a network topology, but it is not always realistic, particularly when you're working with non-modern organisms with a very large genomic data set. So there is a way to find the root position using SiTe patterns without using outgroups for the trees. And we are trying to extend that idea to the networks so we can eliminate the need of outgroups. The second gap is relaxing some assumptions that are not biologically realistic. And one idea is because we are assuming there is a single population size parameter throughout the entire topology, and population size changes over time. So we want to find a way to accommodate, to remove the constraints of having a constant effective population size. And currently, and for this is more of not the method perspective, but more of coding way, we want to accommodate more than a single individual in the species. So currently, we are assuming a single individual is included in the data for the estimation. But thanks to the development of next generation sequencing, it's not very hard to have multiple individuals from a single species. So we want to accommodate that case as well. And finally, we are assuming the true network lies in the class of level 1 networks. And this is nice. This is nice in terms of computational tractability, but it might not always represent the true relationship between the species. So we want to expand our searchable network space beyond the level 1 to accommodate more complex evolutionary histories. So with that, I'd like to thank my co-authors and people who I've been working with in Ohio State, as well as the funding agencies from the institutions that I'm working with, as well as the academic societies, and Solis Labs lab people who I'm having a lot of fun working together, as well as NSF, which helps me to stay in Madison and work in these cool projects. Thank you very much. And happy to take any questions if anybody has. Yeah, yes. Okay, I'll go ahead.
So early in the process, you chose the Jukes Cantor model.
That's right.
Because had desirable properties.
That's right. Yeah.
Simplification of the SiTe patterns. I certainly understand that. But what if the true sequences aren't evolving according to Jukes Cantor patterns? So I wondered if you had done any simulations where you simulated sequences using a different substitution model.
Exactly.
And how to find it.
That's actually a very good point. And because in PhyNEST, we assume the sequences evolve under the Jukes Cantor model, which is probably the simplest model. And the reason that we are using Jukes Cantor is it allows us to compute the true SiTe pattern probabilities in a short time. But in our simulation study, we simulated data under a little more complex model than the Jukes Cantor model. And it turns out to be is performing quite well. It's not actually like crazy complex model. It's actually a little bit more complex than HKY model. But definitely it's worth to look into how the PhyNEST will perform when we simulate data under a very complex model of sequence evolution. So yeah, we tried simulation study under a more complex model. And the good news is it's kind of robust. Yeah.
So I appreciate the kind of gain in computational time, especially when you have a lot of gene trees to estimate. But one of the other major issues is just the number of terminals in a given network and the effective computation time on that. Just Phylogenetic and SNAC, and it kind of maxed out at 20 to 30 terminals.
Yeah, that's right.
Is there any computational gain, or I guess what is the upper limit in terms of PhyNEST in terms of number?
Right. That's a great question. And for the method that uses composite likelihood framework, I'm expecting the computational time will be consistent regardless of the number of loci. But what about we have large number of tips? What about we have large number of sequences? The computational time will go very high. And the number of tips for PhyNEST that I tried was 10 species. And 10 species might sound very small, but it's kind of big for network analysis. And I stopped PhyNEST because it did not finish running after 2 weeks. But at least it works for 7 tips. So for now, we can use this we can use PhyNEST for relatively smaller data sets. So the point is, okay, we are bypassing the gene tree estimation step and then we're using all of the signal. That's good news. But what about the computational time when we have large number of sequences? That is the bottleneck, and that is going to take a lot of time. I hope it's solvable by using more efficient coding, but not really sure if it's going to be solved in a short time. Yes.
And have you worked on removing the level 1 constraints on the network?
In PhyNEST?
Yeah.
Oh, in PhyNEST, not yet. It's a great. So actually, I'm working with Dr. Claudia Solis-Lemus to understand the identifiability of level 2 networks using what's called quartet concordance quartet concordance factors. It's a type of information that you can extract from the set of gene trees. And we are working on that if the parameters are identifiable for level 2 network, but for PhyNEST, not yet. And I think once that project is complete, I think I can use whatever I learned there to apply to PhyNEST using SiTe patterns. Yes.
You always refer to the tips of your tree as species as opposed to taxa.
Right.
I've always said that, try to learn to say taxa instead of species. I'm wondering, you're a zoologist by training, right? So what is your definition of a species?
That's a very, very difficult question. So the question was, what is my definition of a species? Actually, that's the question that I got for my candidacy exam, and we spent an hour talking about that. Yeah, there is no single sentence answer for that. And the reason that I'm using species over taxa is we are assuming species network topology. So we are assuming each tip represents a species. That's why I'm continuously using the term species. But taxa is also a good way to refer a unit in phylogenetic analysis. So taxa is also right. But I thought species is more accurate at least for PhyNEST case. And I know people who are working in this coalescent model refer the tips as populations rather than species as well. So I guess it's just my preference. And I'm so sorry, I don't have a good answer for what is species. Great, thank you very much for listening. And I'll be around, and I'll be in the Madison if you want to try PhyNEST in the future. Thank you.